Yes, sir. Very good afternoon to students, colleagues, and higher officials for online GPAT classes of 23rd day post lunch session. And today I am going to discuss about some important topics under pathophysiology. So this section you will find under the subject human anatomy and physiology as well as pharmacology. Okay, so more the condition questions from pathology will come along with the concepts of human anatomy and physiology as well as pharmacology. So this is very important course, very important subject for and pharmacology. Okay, understanding the concept as well as answering the and choose the right answer. Okay. So myself, Raghuveer Rodda, I am working as an assistant in the Department of Pharmacology of Mother Teresa of Pharmaceutical Education in this Karnataka. Today, under the pathophysiology, I am going to discuss about three important topics. One is cell injury, cellular adaptations, and cell death. And second one is inflammation and cell repair. And third topic is water or fluid electrolyte balance. Maintenance of water or fluid electrolyte balance. Okay. So they are the very huge topics, but I am trying to give very important points which are very helpful to GPAT examination to with a knowledge of anatomy as well as pharmacology. Okay, right. So before going to the details of cell injury, first let us understand what is cell and what is cell injury. So as we know very well, the entire living organisms are composed by basic structures, and that basic structure as well as functional unit. So that basic structural and functional unit of the human body or the living organism, from single cell organism to multicellular organism, from single cell organism to multicellular organism, is nothing but your cell. The cell, as you know very well, it is not only maintaining the structure of the one particular organ by having its place or its location in the tissue. Okay, it is with the help of a group of its organelles, are simply called as with the help of cell organelles. The cell is going to perform its physiological actions. We can call it as a assigned function. Every cell of the body is involved in the performance of certain function to make it viable and to keep it itself in a working condition. So to maintain the viability and to keep itself in the working condition, the cells are going to control the composition of the environment, because the viability will be depend upon many number of factors. It can able to perform its functions when it is present at a suitable environment condition. So to maintain that, it is going to control, the cell is going to actively control the composition of its surrounding environment. So it is not only maintaining or controlling its surrounding environment by various mechanisms, it is also going to maintain intracellular, okay, intracellular malia. So by controlling external environment, it is also control its internal environment. At what? At a narrow range of physical, physiological parameters. So by controlling the various physiological parameters like enzyme levels, like hormonal levels, like ionic levels. So by controlling all, the cell is going to maintain its external environment and the intracellular malia at a narrow range. Because there is a small difference in between the normal condition and abnormal condition, definitely affects the cell functions and the cell structure. So let us see what happened. What happened to the cell if this external environment changes? If this intracellular malia is going to changes? Definitely, when the cell is working with a different type of things and it is encounters the various types of conditions, okay, definitely it is going to undergo a stressful condition. So physiological stress, under physiological stress means when the function of the cell increases. There are certain cases. There are certain cases where the cell is going to work more. The cell is going to perform its action more than the normal to meet the ongoing requirements. For example, suddenly a person starts running from the normal walking position. Okay, all of a sudden the person starts running. At that particular condition, there may be a increased stress on the muscular cells of the lower limbs. Okay, on lower limbs, on the muscles of the lower limbs, the stress may be increased. So at that condition, there is a chance of physiological stress. Because while the muscle contraction is a basic function of the muscle, but what extent they have to contract to produce that kinetics is a very important. And apart from a physiological stress, again is the pathological stimulus. Again is the pathological stimulus, which may be called as again is the injury condition. So keep one thing in your mind: injury is you should not confuse with the wounds. Injury is a condition where it will be characterized by a change in the structure of the cell or change in the function of the cell. So, when the cell encounters a physiological stress, a demand of its original function, and a pathological stimuli, a abnormal, a non-regular thing, the stress by the non-regular things, stress by the injuries, stress by the infections, stress by the presence of foreign substances. So when the cell encounters such problems, so immediately the cell is going to undergo some changes called as adaptations, because the cell is very important. For one particular organ or one particular tissue, the cell is a vital structure. So immediately, against the physiological stress and against the pathological stimuli, it should not become calm. It has to fight or it has to adjust. Okay, before dying. Before dying, it has to adjust to the ongoing situations and ongoing environment conditions or current environment conditions. So such we are called as adaptation. So before going to die, the cell as it is a viable in nature, it is try to adjust to the change environment condition to maintain its viability or to maintain its physiology or to continue its physiological function. So that change environment is called as a new steady state. A new steady state. Okay, the steady state is different and new steady state is different. The steady state is the one in which the cell is having its original structure, or cell is in its original number or original shape, original size, with a balanced balanced fluid content, balanced what we call ionic content and everything. But when that contents are changed in respect to the concentration, with respect to the quantity, with respect to the quality, so that change condition is called as the new steady state. So to achieve that, the cell is going to does certain changes, and that changes will be called as the adaptive changes. So to function normally, to maintain its structure, so to attain a new steady state, okay, it is going to undergo certain thing, okay, which are suitable or which are compatible to change against the change environment condition, and which will be in which condition it is viable in the new environment. Okay, for what reason it is going adaptable? For the adaptations to maintain its viability. It is compatible. The structure of the cell, the internal environment of the cell is compatible with the new steady state. And if the pathological stimulus is increased, if there is an increase in the pathological stimulus, if there is an increased physiological stress more and more, so at that particular condition, okay, if the pathological stimuli or pathological stress or physiological stress is too severe, then the affected cell is going to die. Okay, immediately the cell never dies against the change environment or against the new conditions. Okay, it will withstand up to certain extent. And if that is also goes high, it is not tolerable. The pathological stimulus is not tolerable. Physiological stress is not tolerable by the cell, then immediately it will undergo a type of processes or a stepwise process where the cell is going to die. Then what are the basic causes of this cell injury condition? Which conditions are going to create a change environment condition within the cell, as well as within the cell are intracellular melee, as well as the intermediate external and intermediate environment condition, like hypoxia, the no oxygen condition, and ischemia, the unable to get a solid perfusion based on as per requirements, a chemical agent, the presence of chemical substances, a physical agent like excessive thermal energy, that excessive temperatures, and very solid cold climatic conditions, and infectious organisms and immunological reactions, because you may know immunological reactions are very helpful. They are type of defense mechanisms, but same immunological mechanisms, okay, especially autoimmunity, that such autoimmune immunological reactions are able to produce certain certain cellular injuries. Okay, so when we are going through the details, we'll discuss all them. And another important thing is genetic defects. Genetic defects, the inborn, the genetic defects which are takes place during the life. Okay, will chromosomal abnormalities or mutations. Okay, definitely when we are going to expose for the harmful rays or harmful radiations like UV radiations, sonic radiations, definitely they are going to show their impact on the genetic structures. So if the chromosomal abnormalities or mutations are takes place at the chromosomal level, so definitely that will lead to the various types of injuries conditions and nutritional effects. Okay, when you're not taking a solid diet, like a diet with a proper components like vitamins, then substances, salt, glucose. Okay, all the things required for our body activities. So if you take less, or if you take more, okay, it's not like that. Injury may always takes place in the when you consume less food substances. The injury may takes place even when you take the more nutritional substance. Like for example, your lipid, okay, intake lipids. When you take the huge lipid substances, okay, it leads to the condition called as atherosclerosis. That will be one of the reasons for the condition called as angina pectoris. And aging. Aging is also very important factor where the people are going to reduce some of the biological activities. With the age, there may be loss of some organ activities that may not be able to excrete such chemicals or such endogenous substances which are reactive in nature and which are able to produce cellular injury conditions. Okay, everything. Okay, which may be external or internal, or which may be exogenous or which may be endogenous. With some extent, they are to produce injury in the when they are present in the human body. Then, what are the basic principles? Basic principles present under the by what? Okay, by what mechanism your cautious agents or cell injury causing agents will produce the injurious conditions. So the response of the cell against the cell injury is not an all or nothing phenomenon. So it is not like that during the injurious condition. During the injurious condition, the cell activities are completely abolished or cell activities are compromised. There are no such things. The intention of the cell is that it is going to maintain its structure and it is going to perform its physiological activities. That was the main intention of the cell. So there are no all or nothing. All means entire function, nothing means zero function. There is no such things. It is tried to maintain by having certain intermediate points, by having certain compromising points. The cell is going to produce its response against the injurious condition. And what extent it will give the response? During the cell injury, the response given by the cell or response by the cell against the stimulus or against the injurious agent is completely depend upon what type of injury it is, whether the injury is reversible or irreversible, or whether injury is severe or not too severe, and status of the cell. So at what state the currently cell is present, whether the injured cell is newer or whether the injured cell is already affected, whether the injured cell is in between a process of inflammation condition. So all will matters here. Genetic makeup. Genetic makeup is a very important principle in the which mechanism cell injury caused by the causative agents. The genetic makeup will directly what extent your cell defense against the injurious agent and what extent your cell have protect against the infectious agents. The genetic makeup. That will tell us what extent you have the immunity. In situation, the best example, okay, against the COVID-19. The COVID-19 is producing the different type of things in the humans. Some people are affecting very fastly. Okay, some oldest people are able to recover from that. The people with the age of 30 to 40 years, they are dying against the COVID infection. So genetic makeup is matters. So what extent the genetic makeup of the particular cell or particular individual will defense against the infectious organism, and how its response to a one particular stimulus is very, very important. So the type of the injury and status of the cell and genetic makeup of the injured cells are very, very important. And another important thing is that, so if one cell is injured, will the injury restrict to that particular cell? Is not. It's always not. When one cell of the one tissue is injured, as they have a complex interconnected mechanisms, because the cells of one particular system have a many connections in between them. Okay, there may be a tight junctions, there is a presence of gap junctions, there is a presence of adherent junctions. Okay, there is what there to coordinate one with another, because the function of one particular organ is done by the what are all the cells present in the tissue, which is going to form that particular organ. They have interconnections. They have a communication mechanisms with the help of cytoskeletons, with the help of various ions which are exchanged in between one cell to the another cell, and by having a connections in between the cell, a complex interconnected systems. Okay, so when a single local injury, the injury of the one particular cell may lead to the secondary and tertiary effects. The effects that cell injury effect will be observed on the effect of other cells also, on the performance of other cells also, because the output of the one particular organ is dependent on each cell of that particular organ. Then another important thing, when the function of the cell lasts, will it last the initiation of infection or initiation of injury or starting of injury condition, or the cell function law either at the end of the injury? It's not. So cell will lose its function. Cell will lose its function before losing its biochemical, before losing its biochemical and morphological features. So cell functions are completely abolished if the injury is irreversible type. If the cell injury is irreversible type, definitely the cell functions are lost at the middle. Okay, at the middle, before observing the morphological changes and before observing the biochemical changes, because cell injury, when we can call the cell is got injured, definitely that would be characterized by change in the structure and change in the physiological function. Don't think injury is wound. Wound is completely different. Wound may take place because of cell injury. So when cell is got injured, if stress is increased, and cell will move towards a process called as die. What's a process called as death process? And before reaching that death process, before come to the end of that process, okay, very far, very before to that, cell functions are completely lost. Once cell function lasts, immediately there may be appearance of morphological manifestation, there may be appearance of a biochemical deviations are completely observed. What are the basic biochemical mechanisms? So how the all 8 type of causative agents or causative conditions are approach a procedure to cause a cell injury. So first and most important mechanism will be utilized by any type of injury causing agent is that the loss of energy. So how the cell is basically injured, how cell functions are stops, and how the cell structures are get deformed. Cell structure get deformed. The first and foremost important mechanism by which the cell injury starts is loss of energy. There is a depletion of ATP, because ATP is a chief molecule responsible for energy of a cell. Cell is utilized. Cell is utilized energy which is present in the form of ATP, which is produced in the specified organelles, powerhouses called as mitochondria. So whenever the cell encounters a condition called as ATP depleted conditions or oxygen depleted conditions, immediately, okay, cell encounters or face a one problem called as inhibition of oxidative phosphorylation. Production of energy by the utilization of oxygen and the glucose molecule. Production of energy by the utilization of oxygen molecule and the glucose within the oxysomes of mitochondria. When the cell is unable to get the oxygen, and when the cell is unable to get its perfusion because of any reasons, immediately there is no sufficient amount of oxygen reach to the mitochondria, which are responsible for oxidative phosphorylation on the oxysomes of mitochondria. Definitely, the cell is not going to produce the energy molecule called as ATP. So when the ATP got depleted, the ATP is a very, very vital molecule for the entire range of cellular functions. Cell, what type of functions to be done by the cell? Definitely 90% of cases, the cell needs energy. The cell needs energy, especially the ATP-driven mechanisms. All the ATP-driven mechanisms are required energy in the form of ATP for the active physiological mechanisms. The cell definitely requires a compound called as ATP. So first and foremost thing which are going to shift the cell towards the injurious conditions, shift from normal to the injurious condition, is that the depletion of ATPs, okay, depletion of energy by the improper production of ATP molecules. So ischemia is a very, very common and very, very vital cause for the wide variety of cell injurious conditions. Okay, so no need to infect. So if by any means the cell is unable to get its perfusion through the capillaries, definitely the first and foremost function of the cell is affected is nothing but production of ATP by the oxidative phosphorylation. Aerobic respiration of the cell is get affected. And next important mechanism by which the cell injury is continued, and cell injury is able to produce intracellular variations and the physiological variation is the mitochondrial damage. It is very, very, very important character, a very important mechanism by which the cell gets injury. Mitochondrial damage. As I said, all organelles of a cell, okay, the mitochondria are very, very important, which are powerhouses, okay, which are act to generate a power, which are responsible for wide variety of action. So mitochondria is a double layered matrix structure. It consists of double layer matrix structure. So it is a two-layer structure In the inner folds, in the inner layer folds, there is a presence of a hair-like structure called as oxysomes, which are involved in the electron transport chain mechanisms. Electron transport chain mechanisms where the oxidation process takes place, where the glucose undergo oxidation process to produce the ATP by the oxidative phosphorylation mechanisms. When the cell is encounters a severe stress condition, when cell encounters a severe stress condition, immediately what happened, the calcium concentration in the intracellular, the calcium concentration in the intracellular region, it gets increased. Calcium concentration in the intracellular region will get increased. Okay, so when the intracellular, because of the calcium influx, the calcium from extracellular portions will enter into the intracellular portion. When calcium influx takes place against the physiological or pathological stress, especially during the pathological stress, the intracellular calcium levels are increased, and once the intracellular calcium levels are increased, now whatever the excess of calcium present in the intracellular region now slowly enters into the mitochondria. For example, here there is a mitochondria. So whatever the intracellular calcium increase here, slowly they are try to influx into the mitochondrial membrane. So this is called as permeability transition. It is called a permeability transition, and this permeability transition will cause damage of the mitochondria. So you see, at the end of this particular stage, there is a presence of the vacuoles. Which consists of a type of what is that, chromosomal material. Chromosomal materials are the clumped chromatins. Clumped chromatins are generally occluded in these vacuoles, and in the middle portion, in the um, inner portion where we will find the many number of oxysomes. At that particular portion, there is a presence of calcium salts. There is a presence of calcium crystals. In calcium crystals are amorphous form of calciums, which are generally present in the inner layer or oxysomes area of the mitochondria. So if you take a small section of a cell which is injured and observe it under microscope, so when the cell is injured, so if injured, the mitochondrial cell will shows a presence of many number of vacuoles, which consists of a small fragmented genetic material in the form of vacuoles, or whereas in the middle portions or in the matrix, we are calling in the matrix portion, there is a presence of a calcium crystals. Which are not generally observed in the normal mitochondria. So if you observe in any cellular examination, if you observe this type of condition, immediately it indicates that that cell is under physiological stress or pathological stress, which is injured. And the third, another important mechanism, a loss of calcium homeostasis. Okay, so this is also another important mechanism, because the calcium is a very important ion, which leads to one condition called as exocytosis process. Efflux of the cellular content, leaving of the cell contents from intracellular to extracellular region. Okay, generally calcium intracellular concentrations are maintained at a low levels. Intracellular calcium are maintained at low levels, but the ATP depletion takes place. And when mitochondria is unable to produce the ATPs, what happened? The plasma membrane of the cell. plasma membrane of the cell will lose its permeability, because the plasma membrane is a semi-permeable in nature. Plasma membrane is semi-permeable in nature. It does not allow all the contents. It does not allow all the contents. Even it does not allow the essential contents as and when the cell do not require for their activities. When they do not require for their activity. So the as and when the ATP is not generated, or as and when the cell suffered further energies for the production of various proteins, for the production of various membrane proteins, plasma membrane protein. So what are all the protein activity? Protein activity existed at the plasma membrane level. Protein activity existed at the plasma membrane level are definitely impaired. The plasma membrane lose its semi-permeability. When the plasma membrane lose its semi-permeability by the oxidation of phospholipids of the membrane, by the oxidation of by the denaturing of the proteins of the membrane, or by the improper production of the membrane, what we call membrane pumps. Okay, so definitely all influence the membrane permeability. So one is the calcium pump. Okay, calcium pumps present on the membrane. So the function of the calcium pump is that to maintain the intracellular environment or balanced intracellular environment. So the calcium comes from the extracellular regions. Whatever the calcium comes from extracellular regions will always shift into the extracellular regions, and will always send that intracellular or cytosolic calcium into the sarcoplasmic reticulum, into the mitochondria. Okay, so when the calcium pumps are get affected, when calcium pumps are get affected because of improper product, because of inhibiting the production of proteins which are act as a calcium pump, which are act as a calcium pumps, are calcium ion channels, okay, which are also protein in nature. And when they are not able to produce, and they are not able to constitute a part of the plasma membrane component, so definitely that whatever the calcium which is present in the intracellular environment, this intracellular environment, the entire calcium, because towards the concentration gradient, they will reach from extracellular location to intracellular location. Once the calcium comes into the intracellular location, it will cause many intracellular functions, or initiate many intracellular function, which leads to the cell towards death condition, by activating ATPase. Okay, it will cause decrease of ATP, phospholipase. Okay, phospholipase, it decrease phospholipids and proteases. It is going to convert the proteins into amino acids. Okay, and like endonucleases, it is going to break the nucleic acids, and they will be converted into the nuclear chromatins. Are the nuclear chromatin will get damaged by the endonucleases and other nuclease enzyme substance. So all enzymes are get activated, or all enzymes are get influenced by the intracellular calcium. So the increasing of the intracellular calcium is one of the most dangerous condition, where the calcium homeostasis, the calcium balance is influenced a lot, and which will lead to the cell injury condition. Okay, and another important thing which is going to responsible for the cell injury: defects in plasma membrane permeability. It is also another important feature of the cell. Okay, so if you take the one particular cell, okay, as the cell shows a presence of, okay, many number of vital protein substances, in the form of glycoproteins, and among some. So I think everyone knows about fluid mosaic model. Plasma membrane is a lipid bilayer. So lipids in the form of phospholipids. Okay, they are going to form a double layered structure. Or in the lipid layer, there is a presence of certain floating of protein molecules. So what these protein molecules? These protein molecules, some protein molecules are ion channels, some protein molecules are act as a transporter, some protein molecules are act as a receptors. Okay, so there are two protein molecules of the plasma membrane are very important to maintain the intracellular concentration of ionic substances. They are very responsible for maintenance of intracellular ionic concentration or intracellular environment which is required for the proper function of the cell. One is sodium potassium pump. One is sodium potassium pump, another one is calcium. Okay, so to maintain the normal activities, it is very important to maintain the sodium, concentration high in the extracellular regions. At the same time, it is very important to maintain the calcium concentration high in the intracellular. Calcium concentration high in the sodium concentration high in the extracellular region. Because of that, it is called as a major extracellular cation, and because of potassium concentration high in the intracellular, the potassium is called as major intracellular cation. And these two ions will be permitted from extracellular to intracellular to extracellular by means of a pump called as sodium potassium pump, ATP driven sodium potassium pump. And apart from this, there is a one more channel is present, one more ion channel protein is present, which is going to involve in the transportation of extracellular calcium into intracellular regions. Okay, these are also operated by both are operated by ATPs. Both are operated by ATP, especially the sodium potassium pump. Sodium potassium pump, okay, is operated under the influence of ATP. So as and when the depletion of ATP production and mitochondrial damage, okay, the cell suffer from ATP molecule. Cells suffer from ATP molecule because to activate this pump, if the pump has to send the sodium out from the intercellular region and potassium in from the extracellular region, okay, it should be phosphorylated. It should be phosphorylated. So whatever the energy present with the ATP should be transferred to this particular sodium potassium ATPase pump. Sodium potassium ATPase pump. Whenever the phosphate molecule or because of sodium potassium ATPase molecule, the energy present in the ATP will transfer to this particular molecule. Then the ion channel of that particular sodium potassium ATPase pump is open, and towards the concentration, the sodium move from intracellular to extracellular and extracellular to intracellular. So as the ATPs are lost in the injurious condition, so these pumps are unable to function. The the pumps are going to work in the presence of ATP. ATP-driven pumps are completely lost their activity. So even though the concentration gradients are created, they are unable to move the ionic substances across the plasma membrane to maintain the homeostasis. So such plasma membrane permeability defects takes place, and it will also leads to the injurious condition. That is only because of the loss of energy or depletion of ATP molecule. Then come to the next. Okay, this is very important. Most of the injuries, okay, without involvement of external factors, most of the injuries are caused by the generation of reactive oxygen species, production of reactive oxygen species by the cells of the various organs. So as I said earlier, that's to maintain the viability of the cell or to perform function of the cell is going to produce many number of biochemical reactions. So in some cases the two or three products will be clubbed and will produce one end product, and sometimes the one product will be cleaved off, or one product will be broken into the many parts under the influence of enzymatic substances. So when the cell is performing various biochemical reactions, okay, there is a production of huge number of reactive oxygen species, and huge number of what is huge number of H2O2, hydrogen peroxide, and huge number of hydroxyl ions. All these are having a reactive nature. All these are, as they are not stable molecule. They have a accelerations. And they have a reactive power. When they share their electrons with the other molecules. Okay, they are able to react with. They are able to react with the phospholipids, and they are going to react with the various protein molecules, and they are going to react with various membrane structures, and they are going to react with various intracellular protein substances, especially the free radicals. The free radical damage, we can call it as a oxidative stress. The oxidative stress is a major problem for many number of cellular injuries, sometimes which cannot be even identified at the early stages. Okay, by the time of identification, okay, this reactive oxygen species or these free radicals may cause the severe damage of the organ, which leads to the failure of the organ, which leads to the death of the person as well. So we should be very careful about the reactive oxygen species, the free radicals which are formed against the various biochemical reaction. So that's the reason why we have to take a huge amount of antioxidant substances. They are going to, so the function of antioxidant, they are going to react with the oxygen species. They are going to react with oxygen species and reactive oxygen species and free radicals which are formed by the various biochemical reaction, and inhibit the reacting of these free radicals and reactive oxygen species and reactive hydroxyl ions with the membrane structures or with the cellular structures. So let us take what are the neutralization mechanisms the body have. What type of neutralization reactions body have to overcome that reactive oxygen species or the free radicals? Definitely, it is not possible to maintain or to produce biochemical reactions without forming the free radicals or highly reactive species. Definitely, body to maintain the viability, to maintain the biological reactions, everything definitely they are going to produce a free radicals. So the body itself, to make it in a proper working condition, the cell itself has some neutralization mechanisms. So there are 5 neutralization mechanisms we have. The body has exactly the spontaneous decay, the eliminating of the proton, the eliminating of the proton molecules. Immediately, the free radicals are going to destructed. And the very important mechanism is the superoxide dismutase mechanism. Whenever the reactive oxygen species has come, okay, immediately that will be reacted with, okay, and reacted with the free hydrogen molecule to liberate, okay, H2O2. The one more to liberate the one more free radical, or one more the reactive species called as the hydrogen peroxide. But okay, some extent the reactive oxygen species nature has reduced because of producing oxygen molecules. Okay, so it is not left as such. Again, immediately, as soon as the superoxide dismutase function has done, Superoxide dismutase function has done, which produce a reactive species and a non-reactive species immediately. Okay, so this H2O2, this H2O2 will produce the hydrogen ions and OH ions again. Hydrogen ions and OH ions. That is hydrogen ions and hydroxyl ions. So these are again reactive in nature. These both are again reactive in nature. So immediately, though there is a one more mechanism is existed, that is the glutathione mechanism. Glutathione mechanism. This glutathione will react with the reactive hydroxyl molecules and will produce reactive hydroxyl molecule will produce water. And it is also produced the glutathione disulfide. Okay, it is a glutathione disulfide. So both are non-reactive nature. Glutathione disulfide and water both are non-reactive. So in such way, the superoxide dismutase enzyme and glutathione, okay, it is also one enzyme, okay, which are going to neutralize the reactive species which are formed by the end of various reaction. For if the glutathione is absent, it is not like that. Okay, all cells of human body shows presence of the similar concentration of glutathione. Some cells will not have the glutathione. In such cases, so what are the reactive molecules, that is hydrogen peroxide molecules will produce by the superoxide dismutase mechanism. Immediately they are going to attack by the kettle. They are going to attack by another molecule called as catalase. Another molecule called as the catalase. Okay, whatever the hydrogen peroxide will react with the oxygen molecule, which is rich in blood. Oxygen molecule which is rich in blood and which will be obtained by the blood perfusion. It is even available in the intracellular regions, which is comes from the exchange of carbon dioxide and oxygen. So immediately, whatever the hydrogen peroxide produced by the various reactions of the cell, okay, so this hydrogen peroxide will react with H2O2, okay, which is non-reactive and one is reactive. Immediately, it will be converted in the form of H2O2 will be cleaved out, okay, in the form of catalase. Okay, in the form of catalase, it will produce the oxygen molecules and it will produce the water molecules. Okay, H2O2 will be cleaved out. H2O2 will be cleaved out into oxygen and water. Cleaved out oxygen and water, and immediately, okay, so it is going to reduce the cell itself is try to neutralize all the reactive oxygen species will comes or will produce at the end of various biochemical reactions. Okay, so like that, does every cell have its own type of free radical neutralization mechanisms. So if this will be disturbed by the cell injury. So what are all the reactive species which are produced here, like hydroxyl ions, oxygen reactive species or free reactive molecules. So everyone will produce are going to react with the protein molecules of the plasma membrane or the or the ribosomes which are present in the rough endoplasmic reticulum. So they are going to react and they are going to cleave or they are going to link the different type of protein molecules and will produce the non-functional intracellular substances. So apart from these enzymatic substances, okay, some endogenous and exogenous antioxidant. So just now I said before starting this neutralization mechanism. So against the reactive oxygen species, okay, the things which are responsible or which are very very helpful are nothing but your antioxidants. Okay, the vitamins, vitamin E, vitamin A, vitamin C, okay, and beta carotene, okay, which is rich in the carrots. Okay, vitamin A it is rich in the leafy substances. Vitamin E it is rich in the oils, and vitamin C it is rich in the citrus fruits. Okay, all these substances will helpful to avoid, helpful to neutralize all the free radicals which are going to cause damage to the membrane of the mitochondria, damage to the membrane of the plasma membrane, damage to the membranes of the endoplasmic reticulum and ribosomes and nuclear membranes and everything. So the antioxidants are very very important things, okay, which are going to reduce the injurious condition. So what happens if that free radicals are not killed? If we are not able to regulate, or if the cell do not have the free radical tackling mechanism or free radical neutralization mechanism, so what are your free radicals are going to does, and what will be the impact of the producing free radicals of various biochemical reaction if the cell do not have the adequate neutralization mechanisms? The free radicals which are produced by the various biochemical reaction will try to damage the cell by three mechanisms. One is lipid peroxidation of the membranes. The membrane phospholipids are going to damage, are going to convert it, and the DNA will be fragmented. The nuclear material will get damaged. The nucleosides are going to react with the reactive species, and they are going to lose their bonds, and they are going to lose their structures and the protein cross linkings. So when as the membrane consists of the phospholipids which are polyunsaturated. So whatever the phospholipids which are present in the membrane, which are polyunsaturated, are more vulnerable to the oxygen free radicals. So immediately, so if the intracellular free radicals are not neutralized by any suitable mechanism, immediately they will come and catch the phospholipids which are present. Phospholipids are lipid substances which are present in the membrane and which are mainly responsible for the semi-permeable nature of the cell. Okay, they will get converted, they will get metabolized, and they will get utilized for the liberation of the various products. And DNA fragmentation. So DNA is a very, very important material present in the chromosomes. It's a genetic material. And so whenever these free radicals react with a thymine, it is one type of nucleotides, one type of nucleotide, and especially present in the DNA as well as in the DNA of the mitochondria, or in the DNA of the nucleus. Okay, so that thymine will be get reacted, because the thymine consists of more number of sulfhydryl things, and your free oxygen species will, or the OH radicals will come and react with that particular sulfur group present in the thymine compound, and that will cleave the structure of the thymine, and the DNA will be fragmented. The DNA structures will get disturbed, and it will cause us the breakdown of the double helical structure, and the double strand of DNA into the single strand. When the DNA will convert into a single strand, okay, definitely it will be initiate the other steps like RNA production and protein synthesis, and that too will be formed, that too will be initiated, and which will produce the proteins which do not have any essential role in the maintenance of cell structure as well as in the maintenance of cell function, because whenever DNA will undergo transcription process, and the RNA will undergo a translation process, there will be production of the essential protein substances by the inclusion of amino acids and the ribosomes. So if the DNA will be fragmented in a improper manner by the improper cleaving of the DNA at a different stages, definitely the DNA lose its structure, and whatever the fragments which are formed, they do not have the proper structure and functional regulating activities. And protein cross linking, the formation of a protein with by a linkage of two or three together, which do not have any structural or any functional thing in the one particular process. Okay, so I may go a little faster because we have left with less time. Then, so that are the basic important mechanisms which are going to cause the cell injury. And another important thing is that, okay, the cell injury by the reperfusion. So we should give some importance to this reperfusion, because especially the reperfusion damage is very, very important when the irreversible cell or the reversible cell. Irreversible injured cell, especially the reversible injured cell, okay, is encounters a condition called as reperfusion. If a cells are reversibly injured, if the cells are reversibly injured due to the ischemia, if you if you resort if you resort that reversibly injured cell, if you resort the reversibly injured cell with a very huge very huge rate of perfusion, very huge rate of perfusion against the sudden increase against the sudden increase of perfusion, so the cell damage will be accelerated. So it should be very important when you are tackling with the injured cell. You should not give anything all of a sudden at a high concentration, because instead of repairing that particular cell by, or instead of coming out from the injury by that particular injured cell, the ongoing injury will be accelerated, because at the injurious condition, at the damaged condition, some of the vital activities of the cell are compromised. Some of the vital activity of the cells are compromised, and if you reperfuse, or if you provide all the requirements all of a sudden, at a higher concentration, definitely, okay, will cause accelerated injury instead of recovering from the injury. So reperfusion damage is clinically very very important process, okay, which is going to cause the myocardial as well as cerebral infractions. Okay, so for example, whenever the heart is affected by the ischemia condition, heart encounters a condition called ischemia, improper perfusion. Okay, it will undergo certain injuries condition, which will be characterized by the pain in the chest region. But all of sudden, if that ischemia will be reversed by the perfusion, by the sudden high perfusion. Okay, what happened during the ischemia? Some of the cardiac cell activities are compromised. They are going to contract at a little nature, or they are going to produce little electricities for the contraction and relaxation of the myocardium. But whenever the resuscitation has done, all of a sudden it will increase the blood supply at a huge extent. So against the increased supply of vital element, so in the compromised condition, the cell will suddenly increase the pace of its action. That particular pace will hurt. Sometimes a sudden stroke will come, okay, and it will collapse all the heart myocardial cells. Sudden contraction. They will undergo sudden contraction, and the cell death will take place. Sudden cell death will take place. So the reperfusion, whenever you are working with injury, it is done by the ischemic condition, are done by the hypoxia condition. So you should be very carefully tackled with that. And you have to select the drugs in such a manner that it will be slowly reverse that, and it will slowly take out that cause of the injury. So you should not retake that injury all of a sudden. Okay, it is very very dangerous condition. Okay, and come to the another type of cell injuries. So chemical injury, how chemical injury will take place? Apart from the physiologicals, apart from the aging, apart from the genetical features, so how does the chemicals are going to cause the injurious conditions? So damage of the best example is the mercury chloride damage. Mercury chloride damage done to the various organs. Mercury chloride damage done to the various organs. So in the mercury chloride, okay, so that is sulfhydryl groups. Okay, so mercury chloride will go and react with the sulfhydryl groups. React with the sulfhydryl groups of various amino acids and various proteins. And when mercury chloride, it is a heavy metal, okay, it is going to bind a very strongly with the help of covalent bondings. With the help of covalent bondings or coordination covalent bonding, the mercury will permanently bind to that particular sulfhydryl groups of amino acids or sulfhydryl groups of various protein substances. Okay, where there is no dissociation, and even the metabolism of such resulted groups are not possible by the enzymatic system which has existed in the body. So what are all the proteins which are under the influence of, or which are with the binding of mercury chloride, they will lose their all characters, or they will lose their all structural and functional features. So the proteins even present in the body, but they do not able to produce any function. So they do not going to perform the structural activity or structural maintaining activity or the functioning activity. And another best example to understand is that the damage of the hepatocytes, by the calcium tetrachloride. So it is a very, very classic example. The chemical induced injury. So whenever the carbon tetrachloride is injected at a high concentration, in the body, the carbon tetrachloride will convert it into the carbon trichloride free radicals. Okay, converted into carbon trichloride free radicals, and these carbon trichloride free radicals will go and bind with, okay, triacyl glycerols. Triacyl glycerols of liver cell are which are present in the smooth endoplasmic reticulum, okay, and the membrane phospholipids. Triacyl glycerols and membrane phospholipids, okay, they are going to cause the lipid peroxidation. And they are going to damage the liver parenchymal cells. Okay, what they will do? So whenever the carbon tetrachloride convert into carbon trichloride, okay, reactive species, and this reactive species will go and bind with triacyl glycerols, okay, which are present in the smooth endoplasmic reticulum, or which are present in the liver structures and phospholipids of the your hepatocytes of the liver, and they are going to accelerate peroxidation, lipid peroxidation. They are going to accelerate the lipid peroxidation of the hepatocytes and the smooth endoplasmic reticulum, which are going to release some enzymatic substances again. Okay, they will have digestive properties. Sorry. And that will be finally damage liver cells, which will lead to the condition called as a liver cirrhosis, or a liver necrosis, okay, liver cirrhosis, liver necrosis, and fatty liver conditions, and more type of which is the classic example of your liver injuries. And another important thing is that the liver injuries are also the chemical injuries are also caused by the production of toxic metabolites. So whenever the liver is got injured, and the important character of the liver is that it have a very, very essential role in the biotransformation. So it will convert the compound, the drug molecule, or endogenous molecule into such a molecule which does not cause any harmful to the body, and in which way it is possibly eliminated out of the body. Okay, in case of drug substances, whenever the liver isoenzymes like cytochrome P450 and a group of its enzymes will act against the drug substances, it will convert your drug into the drug metabolite, and which is not harmful to the body. But whenever the liver got damaged, it is not able to produce the liver isoenzymes and the drug metabolitic enzymes, and where the drug will be freely available for the various chemical substances or various injurious chemical substances. And while reacting with the drug substances, the chemicals which are exogenous in nature, and they are going to react with the body, and they are going to biotransform into one particular substance, and which is a toxic metabolite. It is a toxic metabolite of your drug substance, and this toxic metabolite, if it is a heavy and water insoluble, it is a heavy molecule and it is a high molecular weight and it is a lipid soluble in nature, especially lipid soluble in nature. At that particular condition, it will go and deposited into various organs, and this toxic metabolite produced by the drug substances will cause liver injury. So such type of injuries are called as chemical injuries. Then what happened? So as I said, as soon as the liver get injury, as soon as the cell get injury, it will not move to a process called as death. Okay, as it is a very vital structure, very vital component of the human body. Okay, so to maintain its viability, it is try to maintain its structure, at least it is try to maintain its activity by adjusting to the change environment to keep up to produce a new steady state. So that steady state condition is called as cellular adaptation. The cell adaptation towards the injurious condition. So cell adaptation can be induced. This cellular adaptation can be induced and regulated by many number of regulatory steps. Okay, it is not like that. The cell adaptations are observed, are limited to the morphological features of the cell, or structure of the cell. Okay, if they are going to extended up to the receptors, the processing of the receptor, binding of the receptor to the new components, and the signal transduction, initiation of new signals and new communications, new communication patterns by the injured cell, and gene transcription, production of new type of RNA for the production of a specific protein substances which can going to maintain the cell structures, and which is going to maintain the cell functions, and the new protein molecules which are going to produce which are going to act against the injurious agent as well. Okay, it is not like that only the morphological changes are called as adaptations. No, the receptor remodeling of the receptor, or the changing of the receptor binding location, or changing of the receptor ligand molecules. Okay, producing of the new receptor by the injured cell to maintain its viability. Okay, so will be such type of adaptation will be observed, but the common type adaptations which are generally observed by the simple microscopic examinations are nothing but the four important morphological type which are observed in the structural change of the cell, or structural change of the organ. Okay, so are called as atrophy. Okay, these are four are very important, and hypertrophy, and hyperplasia, and metaplasia condition. So these four are called as the major type cellular adaptation mechanisms. Okay, so this image will give clear idea about, okay, so what happened if the cell is under adaptive adjusted condition, by changing its structural and by changing its intracellular components. Okay, atrophy, hypertrophy, and hyperplasia, metaplasia, and sometimes there is a one more important adaptive condition is there, and which will be called as dysplasia condition. So come to the next. What is hypertrophy? So atrophy is nothing but the shrinking in cell size. Atrophy is nothing but the shrinking in cell size. So when the cell is observed, okay, other than its normal size, okay, in that particular condition, the cell is at an adaptive stage is called as atrophy. The cell shrinking, simply called as cell shrinking, okay, by losing the cellular substances. By losing cellular substances is called as atrophy of the cell. And if many number of cells are involved in the atrophic condition, many number of cells are going to shrink by losing its cellular substances, and that condition is called as atrophic condition of the organ. So whenever the more number of cells are involved in the atrophy, more number of cells are involved in the atrophic condition, so definitely when you observe that particular organ under microscope, the entire organ will be observed as a smaller structure than the original. Okay, so what are the reasons, or what are the causes of this atrophy? I'll go a little faster about this four thing, because we have left with very less time, and we have to go very longer again. And causes of atrophy: so decreased workload and decreased pressure, diminishing of the blood supply and diminishing of the nutrients, and loss of endocrine stimulations, and aging. Aging is one of the very, very important. Okay, and what will be the mechanism? What will the mechanism? The mechanisms are not too specific, but during the atrophic condition, the cellular content will be reduced, and sometimes the intracellular portions will shows a presence of autophagic vacuoles. Okay, autophagic vacuoles are generally observed. The presence of some phagocytic phagocyted vesicles we can call, are lipofuscions. Okay, so fusion of structures will appear in the form of globules which contain a lipid structures. Okay, the classic example is brain atrophy. So whenever the persons are in coma, okay, so at that particular condition, the brain do not work normally, our brain activities are completely reduced. Okay, at that particular condition, if you go for the MRI scanning of their brain, so definitely the brain will have observed as a smaller in nature, a smaller in structure, and there's a clear-cut gap is present in between the innermost layer of the skull and outermost layer of the brain. Okay, there is a huge potential gap is observed. That will clearly indicate that it's a brain atrophic condition. Then come to the next one, hypertrophy. So hypertrophy is completely opposite to the atrophic condition. So when there is a increasing in the cell size, and that particular condition is called as hypertrophic condition. So which has happened because of gaining of the cellular substances. Gaining of the cellular substances are going to give new condition called as hypertrophic condition. And like atrophy, when the many cells are involved, many cells of the one organ involved in the hypertrophy, so then organ become hypertrophic organ, where the size of the organ observed in a huge mass. Okay, huge mass. And what were the reasons for this? The reasons of this hypertrophy is nothing but, okay, there is a increased functional demand. So whenever the the demand from the one particular organ, demand of the one particular tissue is increased, constantly there may be a physiological stress on the one particular organ, that will lead to the hypertrophy, size of the cell. So the presence of muscular body is one of the hypertrophy condition. So if you go for continuous gym or continuous exercise, continuous physical work, okay, so the muscles of the body will get the cells of the muscles of your body will get increase in its size, and it will be a muscular body. Okay, so and like that we have increased demand, hypertrophy. Okay, so in another important point is that, okay, hypertrophy is not only limited to the one particular type, not only limited to the normal cell, it is also influenced, or it will also change, or it will also absorb in the phenotypic cells also. So phenotypic cells are such as cells which are produced against the genotypic variation. when one person is try to adjust into the new environment condition. So at that time, they are going to change their genomic structure, change their genomic structure. So that particular thing are called as the phenotypic content, phenotypic cells. So such phenotypic cells are formed, okay, are also influenced by the hypertrophic condition. So it is very important point. The phenotypic cells are also undergo hypertrophic condition. Okay, so we have a many example cardiac hypertrophy and hypertrophy observed in the what is that? Pituitary, okay, gonadotropic cells of the pituitary gland. Okay, at the time of puberty, at the time of puberty, when there is a need to produce gonadotropic hormones, gonadotropin stimulating hormone. So it is very important to increase the size of the gonadotropes of your pituitary gland, which furtherly release huge amount of gonadotrope stimulating hormone like follicle stimulating hormone, luteinizing hormone, which is responsible for the puberty condition. And next is hyperplasia. So this is very important. Hyperplasia is the condition where the number of indigenous cells are increased. Number of indigenous cells are increased in a one particular tissue. Okay, so compared to the normal number of cells in that particular organ, if against the some physiological or pathological condition, if any organ or any tissue will increase their cells in numbers, that particular condition is called as hyperplasia condition. And most of the condition, the hyperplasia is generally observed in the pathological condition. There may be a presence of certain diseases, there is a presence of certain abnormalities. So the basic reason is endocrine stimulation, the endocrine gland stimulation, the hormonal stimulation. Okay, so generally during the during the adrenocorticotropic hormone stimulation. Adrenocorticotropic hormone released excessively by the pituitary gland, by the adrenotropes of the pituitary gland. So so in the intraadrenal glands of the kidney, okay, the adrenal medullary cells or adrenal cortex cells are increased in a higher number, and that will lead to the more release of your adrenaline and norepinephrine. So such conditions are called as when there is an increase in the number of cells, number of similar cells. So that condition is called as hyperplasia. And sometimes this hyperplasia is one of the predisposing condition. Hyperplasia is one of the predisposing condition for the neoplasm or neoplasia. So predisposing means, okay, it is an indication. It is an indication to get cancerous condition. Okay, neoplasia means the production of newer or abnormal number of cells. Production of newer or abnormal number of cells, which is one of the characteristic feature in the cancerous condition. So whenever the number increases, that condition is called as metaplasia condition. Hyperplasia condition. So come to the next one called as metaplasia condition. So metaplasia condition is a reversible condition. It is a reversible condition in which one adult cell type is replaced by another adult cell type. Okay, in one particular tissue, as and when or whenever the cell is have a stress, okay, physiological stress, or cell have a pathological stress to withstand that particular stress condition, to perform their normal duties or to maintain their structures, okay, it is going to produce a better cellular structure. It is going to produce a better cellular structure, which is going to produce more defense, against the stressful condition. So when one type of the cell, one adult cell type is replaced by another type, so you should not consider with cell proliferations. So because in the cell proliferation, okay, one type of blast cell will convert into the class cells will convert into the again into the class cells again, or finally into the normal cell of the one particular neuron. So there will be a clear cut difference in the structure of the cell. So you should not consider cell proliferation as a metaplastic condition. Okay, so because of this reason, we are using the word adult type. One cell adult type is replaced by another adult type cell. At that particular condition, the condition is called as, or adaptation is called as metaplastic condition. Adaptation is called as metaplastic condition. So metaplasia in a indigenous cells is replaced by that of a better cell, which is going to tolerate a specific abnormal condition. The best example is that, okay, intestinal metaplasia of the esophagus. Intestinal metaplasia of the esophagus is one of the best condition to explain metaplasia condition, where the esophageal squamous cells, are going to change. Okay, so generally, if you see the mucous membrane of the esophagus, it is a squamous epithelium layer. So whenever whenever the person is experienced with, okay, GRD, gastroesophageal reflex disease, in such particular condition, under the influence of that gastric stress or gastroesophageal reflex stress, okay, so the squamous type of the esophagus will be replaced by gallbladder cell type, which are mucus secreting cells. Gallbladder cells are mucus secreting cells, and esophagus will finally absorb like a intestine. So how the intestinal mucosa observed under microscope? Intestinal mucosa layer observed under microscope. So whenever the person suffering with intestinal metaplastic esophagus, so that particular person esophagus is observed in the form of intestine. Okay, so in the form of small intestine, because the small intestine have the gallbladder cells in their layer, but esophagus do not have that gallbladder cell. Generally, esophagus contain only squamous or the fish-like flattened dead cell that will be replaced against the acidic stress or against the esophageal reflex stress. So gallbladder cells, there is a conversion of the flattened dead cell into a gallbladder cell. So that particular gallbladder cell will try to produce more amount of mucus to reduce whatever the irritation which is observed in the esophageal irritation observed in the esophageal tract. Okay, so this is about your cellular adaptation, which is undergone by one cell which is encounters a injurious condition or injurious stimulus. Okay, and apart from these four, there is one more condition called as dysplasia. Okay, so dysplasia condition is almost similar to the your hyperplasia condition, where there is an increase in the cell number, but the arrangement of the cell or cell structure will be differ. So cellular arrangement, how the cells are arranged in one particular organ. So there is an abnormality observed in the architecture of that particular cellular layer. So that whenever such condition, such abnormal placement of the cell or abnormal architecture of the cell are present in an increased number, against the increasing number. Okay, so similar cells, but they have a different architecture, abnormal in the arrangement. So such conditions are called as, or such particular adaptations are called as dysplasia conditions. Okay, so that you have to very careful. Metaplasia is different from dysplasia. Dysplasia is similar to hyperplasia, but different from hyperplasia and hyperplasia and dysplasia is that, okay, in the hyperplasia there may be a specific architecture, but the cell number increase. But here also cell number increase, but architecture is different. Architecture is a abnormal. Okay, then come to the another small concept called as pathologic calcification. Pathologic calcification of the cells or calcification of the hormones. So during the injurious conditions, so as I said, the one of the important mechanism is that the calcium influx. The movement of the calcium from extracellular environment to the intracellular environment. So what happened to this particular calcium? And if it is deposited, if that calcification, increased calcium concentration, if it is take place in the diseased or in the abnormal, in the dead cells or in the dying tissue, so what it is called? And if it is takes place, if calcium deposition takes place in the normal cell, so what it called as? So dystrophic calcification, very important, very important for your GPAT examination. Okay, so I in the 99 gate exam, okay, this was the question. Okay, so you should be very careful about such things. So you should know each and every simple concept of the pathophysiology, because it's a very vital bridge in between HAP and pharmacology. So the dystrophic calcification. Dystrophic calcification is a abnormal deposition of the calcium phosphate. Okay, so whenever there is a deposition of your calcium in the form of calcium phosphate in the dead cell or in the dying tissue, the cell which is already died and the cell which is in the process of dying, okay, if the anything happen, if calcium is calcium get deposited in such particular things, okay, so that particular condition is called as dystrophic calcification. And dystrophic calcification is one of the important reason for one of the pathological condition called as atherosclerotic disease or valvular heart disease. Okay, so during the against the your what do you call it? Against the huge fatty substances, and against the obesity. Okay, whatever the fat present, the excess of triglyceride or excess of massive fat present in the blood vessels or in the blood, it is gone deposited over the okay blood vessel which is under stress. Blood vessel cell, it is under stress. Okay, so the cells which are died. Okay, so if the plaque, okay, atherogenic plaque will takes place for that particular cell, it is going to lead to the condition called as atherosclerosis. The narrowing of the blood vessel due to the deposition of the lipid substances and died endothelium cells. And in the walls of the heart, when the heart encounters the angina pectoris condition and hypoxia conditions and your ischemic conditions are myocardial infarcted condition. So that whatever the calcium present in the blood will start depositing on the necro tissue of the myocardium, or necro tissue of the walls of the myocardium layer, and that will cause the valvular heart disease. So whenever the calcium deposited in the dead cells or calcium move from extracellular to the dead cells, that will be called as dystrophic calcification. Whereas the same calcium during the hypercalcemia, whenever there is a more calcium concentration in the blood or more calcium concentration in the body fluids, in that particular condition, what are the excessive calcium present in the blood or present in the body fluids will go and deposit in the normal tissue. The tissues which are working normally or which are healthy. So if that deposition takes place in such cases, in such normal tissues, that will be called as metastatic calcification. And the conditions like vitamin D disorders, vitamin D disorders, and the bone destructions. Okay, bone resorption. We can call it as bone resorption. In that particular condition, the bone will be resorted, or bone will be distracted by the various things, and what are the calcium present in the bone will come to the fluids, and will come to the extracellular fluids, and from the extracellular fluid it will go and deposit in the normal tissues. Okay, bone resorption. Okay, and renal failure, when the kidneys are unable to produce a specific substance, specific substances. Okay, and especially parathyroid hormone, when there is a parathyroid hormone levels are decreased, which is very important hormone, very important hormone involved in the comes from the parathyroid gland, and it is influence the hormone called as calcitonin, which is responsible for calcification process. So when there is a imbalance or occurred in the parathyroid hormone, or increasing the parathyroid hormone levels and such things, then such condition will lead to the renal failure, and which also causes the more and more amount of deposition of calcium in the fluids of the body, and that will be migrated to the normal cells. Such things are called as metastatic calcification condition. Yes. So yes, it does everything. The cell is try to maintain its structure. Cell is try to overcome all the stress, but what are the mechanisms it is initiated? What are the adaptive condition it is produced to fight against the ongoing stress, but the stress is constantly increasing. The stress on the cell is constantly increasing, and the cell injury is progressed. What happened to that particular injured cell? Because the cell is a very teeny structure, it can withstand, it can does its adaptation, and adaptations will help the cell up to certain extent, not to a complete extent. So at a one particular point of time, if stress is not cleared, or if the stress is not removed, especially pathological stress is not removed on the cells, definitely cell lose its functionality, and it will move to the process called as cell death. And the normal cell which is under stressful condition, and which is injured against the various causative organism, based upon the extent of damage done, and based upon causative agent involved. Okay, the injured cell move to the two directions. Okay, to move to the two parts towards the death of that particular affected cell. So one pathway is called as apoptosis. And another pathway is called as necrosis. So the injured cell, reversibly injured cell, when the injury reached the point called as point of no return. So if they will reach to the condition point of no return, so the cell, even the reperfusion or even the stimulus is removed after the point, when cell reach the point of no return, the cell loses all its vital content, and it is not going to regain its structures by the existed cellular content. So find the irreversibly injured cell, which is how at the point of no return stage, will finally move towards the towards the death. The death will take place by the two ways: one by the necrosis, one by the apoptosis. Here, the necrosis is a pathological type cell death. Whereas apoptosis is a pathological type as well as physiological type cell death, because apoptosis itself, it is called as a programmed cell death. Whenever the one of the tissue of the organ fails, the cells, the newly formed cells, are no more required for their activity. So immediately the whatever the new cells produce in the that particular organ will initiate such a self destruction program. a well constructed self-destructed program called as apoptosis. But whereas the pathological, the cell injury have the infectious origin. Injury have infectious origin. Or injury have very huge stress, huge radiation stress, okay, huge radiation or huge thermal stress, then it will do not follow a systemic pattern. Do not follow a systemic pattern of the death process, and that will be called as necrosis. Okay, so we'll see what are the systemic process, and so here, just I kept some of the important information for you. Difference between coagulative necrosis and apoptosis. Okay, so the stimulus for coagulative necrosis is a hypoxia condition. As I said, just now it is a pathological type and it is a physiological and pathological type. So hypoxia and taxins, which are the pathological background, and pathological and physiological and pathological background. And histological appearance, so whenever the cell is under necrotic pattern, so it will shows a swelling of the cellular components and coagulation of the various intracellular materials, necrosis, okay, that is formation of some thread-like structures, okay, fragmented structures, okay, disruption of organelles. What are organelles are observed in the form of a small tiny vessel-like vascular structures, and condensation, presence of a apoptotic bodies. So whatever the intracellular materials are there, even chromatins, nuclear materials, everything are present in the form of, are enclosed in the form of small vacuolar structures, and the DNA will be break down randomly. They do not have any specific enzyme to break down that, and they will diffuse. Sometimes nucleases are attacked very hugely. The DNA will be completely broken down and completely diffused into the whatever the intracellular cytoplasmic content remain. And whereas in case of apoptosis, it is a intranuclear nucleosomal. Okay, it will react with it will react by nucleosomes, and it will be engulfed by the one of the membrane-like structures. And the mechanism for the necrosis: ATP depletion, membrane injury, and free radical damage. And whereas apoptosis condition, there may be a initiation of genes. Okay, new genes are get activated, and new genes are get reacted and get comes into the expression region. Okay, they will produce endonucleases, they will produce set of proteases, and everything they will produce, and they may kill the cell, and the cell kills itself. It is one type of suicide, kills itself, okay, by following a specific pattern. Okay, so tissue reactions, there may be huge inflammatory reactions, there may be involvement of inflammatory mediators there, and whereas in case of apoptosis, okay, there is a no inflammation, okay, in case of it is a physiological, okay, because there is a presence of phagocytosis. There is a presence of one clearance process called as phagocytosis, the cell eating mechanism of the various small bodies which are formed during the cell injury condition. And this will give about the detailed information about the apoptotic pattern, because compared to the necrosis, the apoptotic patterns are very, very important, because it does have a physiological background, as well as it have a background of pathological as well. Okay, so the compounds, like as I said, it is a physiological. So physiological requires the self-destruction. Physiological requires a self-destruction process. So there will be the involvement of some immunological cell, like cell toxic cells, and there may be a presence of a receptor ligand interactions, which are going to send some of the digestive enzymes into the cell, some of the hormonal substances into the cell, which are going to react with nucleus, and which are going to react with DNA to produce the specific RNAs, to produce a specific group of protein molecules, which are going to initiate a process of apoptotic process. And there may be withdrawal of various growth factors and cell cycle regulation, like bulk C, bulk XL, Okay, and there may be a chance of injury. Okay, so apoptosis is also takes place even in the injuries condition. Apart from the physiological, when the body doesn't need their cells, or when organ doesn't needs those particular cells for their activity, for their functionality. Okay, so apart from that, so whenever injury, radiation, toxins, free radicals, so if all things will come and they are going to initiate a capsizes, they are going to initiate, okay, there is a one type of process called as the production of the capsizes, production of capsizes. So these are a group of enzymatic substances. These are a group of enzymatic substances or a group of protease enzymes, a group of protease enzymes which are going to initiate a cell a programmed cell destruction process or a programmed cell destruction process. So all the capsizes will come like cysteine aspartate proteases, cysteine dependent aspartate proteases and direct proteases. Okay, all the protein digesting hormonal substances, sorry, enzymatic substances will come or will produce into the cell by the DNAs and other things, and that will cause the fragmentation of DNA, and that will cause the digestion of the chromatins, that will cause digestion of the ribosomes, that will cause the release of your lysosomal enzymes from the cell which is injured or which is not required for the activities, and finally the engulfing of the that particular digested material in the form of vacuoles, and that vacuoles will detaches from the cells, and finally that cells will be okay will be converted in the form of apoptotic bodies. So apoptotic bodies are looks like a cell, but the material present in it are not in a functional. The cellular organelles which are present in it are very less and they do not in a functional manner. And finally, these apoptotic bodies are engulfed by the phagocyte, like macrophages. They will be eaten by the macrophages. Okay, so this is about cell injury and cellular adaptations and and the cell death. Okay, so now let us discuss some questions. So let us discuss some questions related to the cell injury. Okay, so after this we'll go to the inflammatory process again. So let us discuss very fastly the principal structural component of the cell wall. The principal structural component of the cell wall in bacteria is made up of. Okay, so basically we'll get this type of questions when we are dealing with pathological types. Okay, because the bacteria has a major things are major organelles which are involved in the cell injuries. So in the bacterial cell wall, there is a one principal component is present. Okay, so that is nothing but your peptido, peptidoglycan polymer. So because of this reason, okay, so your components like antibiotics, the drugs like antibiotics are very useful component to combat various diseases of the bacteria. Peptidoglycans are okay, very important component present in the cell. Simple proteins, definitely if it is simple protein, so cell will easily kill. The bacteria will be killed by the osmotic process of the human body. Then complex polysaccharides. So polysaccharide itself are not make the bacteria more rigid against the defense conditions exist in the human body. And glycoprotein. The glycoproteins are present in the bacterial membrane as well as in the human membrane. Okay, it is have some transportation activities. Okay, so peptidoglycans are the very important feature of the cell wall of the bacteria. And come to the next one, dysplasia. So just now I mentioned when I'm talking about dysplasia, many people will get confusion while talking about dysplasia. Okay, so dysplasia is similar to the hyperplasia condition. Similar to hyperplasia condition, there is a difference in between dysplasia and hyperplasia. Is that in the hyperplasia, the cell number will increase. In the hyperplasia, cell number will increase, and in the dysplasia also cell number will increase. But pattern of arrangement, architecture, we can call it as a architecture. The pattern of arrangement of the cell is different. So change in the morphology, no. Okay, cell have their indigenous structure, and change in the cell type, no. If it is change in the cell type, it is metaplasia. Change in the cell type is metaplasia. Okay, change in cell size. Change in cell size, it is hypertrophy or atrophy. So change in cell size, there is no clear whether change in the shrinking or change cell size reduction or cell size increase, but change in the cell size, which may be atrophy or which may be hypertrophy. So where the dysplasia belongs to change in the cell number. Okay, for example, along with cell number, if any among the multiple choice there is a presence of, okay, increase cell number and change in number and improper arrangement of the cell. Okay, change in number and improper arrangement of the cell, then you have to select that one instead of change in number, because there is a change is observed, along with the change there is a observance of improper architecture, abnormal architecture. Okay, so which of the following pigments are involved in the free radical injury? Okay, so there are certain pigments of the human body are involved in the damage of the cells also. Okay, so there is a many number of pigments are observed, but the pigments look like lipofuscin, right? Lipofuscin, it is a one of the compound. Lipofuscin is one of the compound produced by the metabolism lipid cell produced by the lipid metabolism. Okay, and fusion of some lipid molecules. Okay, and which is yellowish, okay, which is yellowish brown color in nature, which is yellowish brown color compound, and it is going to react with, it is going to have some the free radicals. It is going to have some the free radical acceleration property, and that is going to be involved in the pigment based radical free radical injury. Melanin, it is a skin pigment, and bilirubin, it is a product formed by the liver cells. Okay, biliverdin will be converted into the bilirubin by the various enzymatic action, and hematin is a breakdown product of RBC. When the iron of the RBC or iron of the hemoglobin, especially iron of the hemoglobin will undergo oxidation. It will produce a black colored compound. Okay, blackish brown color compound called as hematin. Okay, then the right answer is lipofuscin. Lipofuscin is a pigment which is able to cause a free radical injury. And come to the another thing is hypertrophy. Hypertrophy is due to, so the reason behind the hypertrophy of the cell, increase the size of the cell. Increase size of the cell is because of increased glycogen content. So whenever the work of the cell is get increased, workload of the cell get increased, okay, it is going to need more and more amount of energy. In such cases, okay, so the atrophy, so the cell is to get the more and more amount of glycogen content, for the production of energy, and which will be utilized for the increased activities. So when glycogen content is increased, okay, against the increased activity, or to compensate the increased activity, then to accommodate that, the size of the cell will get increase. So increase protein content, definitely no. Protein content increase means definitely it will be converted as an enzyme or converted as a amino acid or converted into a one of the protein of the plasma membrane. So there is no need to increase the cytoplasmic content or increase glucose content. So glucose content is increased. So definitely it will be utilized for the biochemical reaction, and definitely whatever the glucose comes, it will be readily utilized. And vitamin, there is no relation between vitamin and other things. Then in hypoxia, the cell injury. Okay, in hypoxia cell injury, cell swelling occurs because of increased intracellular. So as I said, the major step in the change in the intracytosolic levels during the cell injury condition. Okay, during the cell injury condition is that, okay, because of change of membrane permeability, so definitely more and more amount of sodium will comes in, and whenever the intracellular sodium concentration increases, definitely that will drag, okay, to maintain osmotic balance. So it will drag more and more amount of the water from the extracellular region whenever cytosolic sodium concentration increases. So the water concentration will increase inside the cell. So during the hypoxia cell injury, more water will come to the intracellular, which is main reason behind the cell swelling. So whenever swelling comes, it will clearly indicates that there is a involvement of liquid compound. There is an involvement of liquid material. And next to that, which of the following is not a feature of the reversible cell injury? Reversible cell injury. So cell swelling. So definitely the cell swelling is a primary step in the any type of cell injury. Okay, so that is because of impairment of what we call the pumps which are going to exchange sodium and potassium, and reduction of ATP synthesis. So reduction of ATP synthesis is the first step in the reversible cell injury condition, a basic mechanism, okay, in the reversible cell injury condition. And reduce cellular pH. So again is the abolishing all the cellular activities, and especially when ATP production is stopped by the aerobic respiration. Immediately the cell will switch to the anaerobic respiration by utilizing glycogen content, which will be converted into the pyruvate by the anaerobic respiration. And when glycogen is utilized for the production of energy, okay, there is a production of lactic acid. And that lactic acid is responsible for increased pH, okay, increased acidic pH in the cell injury. Okay, so that reduced pH. Okay, reduced pH is also reduced pH means towards acidic conditions. Okay, reduced pH means, so when pH is reduced means goes behind 7.4, goes behind 7 like 6.5. So the lactic acid accumulation will reduce the pH, and pH will be maintained at the acidic side. So these are the all normal causes. Defects in cell membrane. Defects in cell membrane is the right answer. So if the cell membrane is get affected, other than the protein molecules, so like phospholipids of the membrane are get affected, so definitely the more and more amount of the water will come, and it will abolish, or it may cause the destruction of the membrane or the bursting of the cell. Okay, so until or unless the cell will retain its membrane integrity, or cell components are not destructed. Okay, so it definitely if you remove the stress condition, or if you remove the whatever the stress responsible for the injury, so definitely cell will recover from that particular injury. But if membrane get damaged, it will generally occur at the last. Membrane get damaged, it will generally occur at the last, definitely the cell will move to the irreversible conditions. Okay, so next to that, come to the next question. Which of the following is a feature of apoptosis? Okay, cell swelling. So cell swelling is even occur in the necrosis conditions. Cell swelling is even occur in the adaptive conditions also. Involvement of large number of cells. Okay, so apoptosis may be observed in the number of cells, as well as apoptosis is also observed in the single cell. When the organ needs that single cell is not required for its activity, it will initiate certain regulatory activities in that particular cells by the regulating compound, by cell cycle regulating compounds like TNF alpha, macrophages, and platelet derived factors, growth factors, and it will move that particular cell towards the apoptosis condition. So single cell, there is no need to involvement of the large number of cells and associated inflammatory changes. Okay, so if there is a, as I just now in my previous slide, I said the difference between necrosis and apoptosis. Okay, so necrosis is more possible more possible by the inflammation. At the same time, apoptosis is because of pathogenic type. Definitely, if apoptosis is a pathogenic type, definitely there is involvement of inflammatory changes. And the feature which is related to apoptosis is chromatin condensation. So whatever the chromatins are there, the chromatins are there in the nucleus, okay, will be come out by the nucleages, by the endonucleages. It will break down at the disulfide bonds. It will break down by the nucleages and endonucleages, and that will be converted into the small fragments or condensed. Okay, so all the chromatin material are chromatids which are present in the nucleus will form a massy structure. So the important feature. So when I'm talking about apoptosis, even I said how the condensation takes place, how the capsizes, the proteases, endonucleases are responsible for the digestion of the nucleotides and nucleosides. Okay, and convert it into DNA into fragments and the genetic material into fragments and a compressed material. So the important feature, the chromosome condensation or chromatin condensation is very, very important feature of apoptosis, and which will not be generally observed in the necrotic condition, because all digestive enzymes come from the endogenous compounds and come from the infectious organisms will completely digest and completely what we call convert that DNA material into a liquid material. So where there is a complete disappearance of chromatins or genetic material appear, but whereas in the apoptosis, which will be condensed and it will be engulfed into the small apoptotic bodies. And come to the last one, with regard to the mechanism of cellular adaptation, which of the following statement is true? Hyperplasia refers to increase in cell size. Okay, hyperplasia refers to increase in cell size. Yes, hyperplasia is a condition where cell size is increased because it is completely opposite to the atrophy. Okay, hyperplasia increase in cell it is not hyperplasia. It is a hypertrophy. Okay, so here it is hypertrophy. It is referred to increase in cell size. Then TNF alpha act as inhibitor of hyperplasia. TNF alpha is inhibitor of hyperplasia. Definitely, TNF alpha is a cell cycle regulating compound. Okay, so definitely whenever TNF alpha will come, so definitely it is going to inhibit the hyperplasia condition. Okay, increase cell size. Okay, it is then increasing what is called hyperplasia, cell number. Okay, cell number. Then metaplasia is a reversible. So when I'm talking about metaplasia, first and foremost thing I said is the metaplasia is a reversible one. So then metaplasia is a squamous. Metaplasia is a squamous to columnar epithelium. Metaplasia is the squamous to columnar epithelium in a distal esophagus. Yes, in the metaplasia. So the one which I used, the example which I used, okay, that will be related to the metaplasia condition, intestinal, okay, metaplasia of the esophagus, where the squamous, the fish type or flattened squamous epithelium cells are converted into the columnar type epithelial cells called as goblet cell. Okay, so here there is one small correction. It is hypertrophy. Hypertrophy refers to, and hyperplasia is not referred to. With regard to the mechanism of cell adaptation, which of the following statement is true? Yes, hyperplasia is related to the cell type. Okay, one cell adult type. So hyperplasia never. Yes, it is correct only. Hyperplasia will never belongs to the cell size. Never belongs to cell size. Okay, hyperplasia refers to the cell adult type, not cell size. Cell size is hypertrophy or atrophy. And TNF, okay, so TNF always acts as a inhibitor. So it is TNF, it is a regulator. It is a regulator, cell cycle regulator. It is always activate the cell cycle. Okay, so metaplasia is reversible. So only squamous to columnar epithelium is a distal esophagus. Okay, so this is about some common bits. Okay, so you should be very careful while dealing with this type of bits. Okay, so it will give more confusion. So you have to read the question very properly. So you have to read the statements very properly. Okay, myself, just for a minute of time, I also get confused with the hyperplasia and hypertrophy. So you should be very careful. So you should read and understand the statements very carefully. Okay, so I'll extend my class up for another 20 minutes to complete the topic called as inflammation and repair. Anyhow, I will go a bit faster to wind this class within the scheduled time. Okay. Then let us go a little faster. So inflammation is defined as the local response of a living mammalian tissue to the injury. So as I said, okay, the injury is not a all or none response. There is an intermediate points also. There is some compromising areas also. So as the cell is a vital thing, so the cell is try to be as a active and be as a functional structure. So to overcome the stress, to remove the stress, the cell will follow certain defense mechanisms, and that defense mechanism, the body immune defense, the body immune system response against to a irritant, against to a injurious agent, or against to a one irritant. Okay, so such particular process is called as inflammation or inflammatory response. The inflammation is a response given by the affected cell, or given by the immune cells of a body, against the injury, or against the injurious organism, or against the irritant, or against the stress. Okay, to what we call the stress, to overcome the injurious condition, or to regain the normal structure or normal condition of a cell. So what are the causes of this injurious condition? So this inflammatory condition. So the causes are mainly infectious agents like bacteria, virus, and toxins, fungi, parasites. Okay, whenever the injury is having a pathological background, and it is a diseased condition type of injury, and the immunological agents like the inflammation may take place because of our own antigen antibody reactions, especially during the inflammation takes place during the autoimmune conditions. Then physical agents like heat, okay, cold, radiation, mechanical trauma, trauma belongs to the accidents and belongs to the various physical stress conditions, and inflammation belongs to the organic substances like toxins, excretory chemicals of the bacteria, like your pyrogenic materials, okay, and inert materials such as foreign bodies like toxins, the materials which are present in the foodstuffs, okay, are also able to produce the inflammatory responses. And how we can identify there is a presence of inflammation, how we can understood the inflammatory area from the non-inflammatory area. So because of infiltration of certain inflammatory mediators and infiltration of the blood contents to the affected area, there is a presence of certain specific things at that particular area. So there is a 5 cardinal signs which are basically observed in the inflammatory condition. One is called as rubor, okay, the redness at the affected area, and tumor, there is a swelling at the affected area, and color, there is a heat, there is a generation of heat. Okay, so there is a because of pulling of the blood to that particular affected area, and when there is a need of migration of the immune cells, there is a need of migration of the defense cells against the affected organism to survive the host or to survive the host cell. Okay, and that will be there will be heat will be generally observed at that place, and there is a huge heat will be present at that particular inflamed area. And dolor, there is a severe pain because of sensitizing of the nerves or sensitizing of the cell by the various inflammatory mediators, and functionalisia, loss of function of that particular inflamed organ. So all these 5 signs are very vital, and these may be present in the inflammatory conditions. All are at the time, or any one of these signs at that one particular time. Okay, it is not like that during the inflammation you can observe all these things. Sometimes redness may be present, redness may not be present. There is a pain and heat, and sometimes there is no swelling, but there is a presence of pain in that inflamed, the pain that particular affected area. So the presence of one or presence of all or presence of any 2 or 3 together will definitely indicate a inflammatory conditions. So based upon the type and based upon the capacity of the host cell to defend against the injurious agent, against the ongoing conditions, or against the stimulus, and what extent that inflammation as encountered by the host cell. Inflammation as encountered by the host cell, there is a presence of 2 type of inflammatory condition. One will be called as acute or innate type inflammatory response, and chronic type inflammatory condition. So both will be depends upon what type of immune action or immune reaction produced against the ongoing injurious condition. So that will decide whether the inflammation is acute type of inflammation or whether inflammation is a chronic type of inflammation condition. One will be because of innate immunity, the immunity which produce all of a sudden against the presence of one antigen or presence of one new antigen in the body, and the adaptive immunity which is done by the which is produced by the antibodies which have already existed in the body, are already when the acute inflammation will fail to combat the ongoing effect of the injurious organism, or ongoing effect of the ongoing stimulus which is responsible for the inflammation. So inflammation is a protected activity. So acute, okay, so acute inflammations are starts. Acute inflammation. So these are the some type of some images which can give idea about the acute type of inflammations and chronic type of inflammation. So the redness, the swelling, okay, swelling is a very common type of signs will be observed in the acute type, which is against the innate response, the response of the immunity by producing antibody against the new antigen. Okay, non-specific antibodies against the new antigen which is entered in the body. Whereas chronic type of inflammation, so there is a huge edema, there is a sometimes there will be deformation. The inflammatory cells, inflammatory mediators or inflammatory cells, when they are acting against the organism or against the pathological organism or against the cause which is behind the inflammation, okay, when they are trying to protect that, they are going to form one deformity in the end organ, that will be stays within the rest of the life. So compared to the acute, the chronic is forming some deformities in the organs, but compared to the chronic, acute are very, very dangerous in nature. So let us see what happened in acute inflammation. So the causes of, as I said, infection and physical, chemical agents, everything are responsible for acute inflammation. So whenever acute inflammations takes place, whenever there is a involvement of antigen and there is a involvement of non-specific antibody. There is an involvement of non-specific antibody to combat the effect of the newly entered antigen. The combat effect of newly entered antigen. So this will be done in the 4 stages. One is vasodilation, one is increased vascular permeability, and one is movement of white blood cells. So these are the protective cells. Okay, these are the defense cells which are present in the body, which is like neutrophils, basophils, eosinophils, okay, granular cells, agranulocytes. Okay, so the migration of WBC to the affected areas, especially where antigens are present in the affected areas from the blood vessel into the soft tissues at the site of inflammation is very, very important step in the acute inflammation. So as the vasodilation takes place, so during the acute inflammation, the first step is vasodilation. So vasodilation is only responsible for the starting of inflammatory process and starting of swelling process by the increasing of vascular permeability by producing a gap in between the endothelium cells which are very closer in the normal condition. So as and when the antigen comes, and if antigen comes because of various inflammatory response initiations, so suddenly the local hormones will release like histamine, prostacyclins, and the nitric acid will release. So all these cells will go and bind to the specific receptor present on the endothelium cells, and they are going to cause the dilation. They are going to cause the dilation of the blood vessel. It is called as vasodilation, or swelling of the blood vessel called as vasodilation, where the more amount of blood will be pulled to that particular dilated area. So when more amount of blood will be poured, and more amount of the blood cells will reach that particular area, and more amount of systemic mediators and inflammatory mediators will move to that particular infected locations or affected location to recover that particular affected area, to kill that particular antigen response for that particular damaged condition, and increase vascular permeability. So vascular permeability is very, very important. So blood vessels are not like other membrane of a plasma membrane of a cell. Okay, so in the blood vessels, okay, the blood vessel layers are formed by endothelium cells, and endothelium cells are observed very compactly. Okay, they do not give the space even to passing of the water simply by the free gaps of water. Sorry, the water gap present in the endothelium cell, because they are very compacted. That makes the formation of a blood-brain barrier, blood-placental barrier, and blood-testis barrier. We do not allow the compounds, allow the toxic substances which are easily entered into the systemic circulation. Okay, so that even other than the blood-brain barriers, blood-placental barriers, and the blood-testis barriers, one of the vital activity produced by the vasculature is that because of compact endothelial cell arrangement, so they do not easily pass the blood materials filtered into the extracellular or interstitial spaces, and entering of the interstitial material even into the blood. But because of histamine, because of bradykinin, These are called as local hormones. And TNF alpha, it is a cell cycle regulating compound. Interleukins and leukotrienes, these are the eicosanoid cells, are the cells which are produced by eicosanoid activity, which are get infiltrated, which are get causes the really extend the space in between the two endothelial cells, like LTC4, LTD4, LTC4, all the the pushing of the blood to the dilated area. This will increase in number, and they will bind to the specific receptor, and they will change the cell cycles of the endothelium cells, and they produce some space in between the endothelial cells, which allows the movement of the blood vessels and movement of leaking of the white blood vessels into the affected area, leaking of into the soft tissues which is affected, and other cells which are responsible for the inflammation will finally enter into that particular affected place. Okay, so what your acute inflammation results? So acute inflammation results, the acute inflammation will move the affected cell to the resolution, complete retaining of the tissue to the normal condition, complete retaining of the affected cell to the normal condition, healing. If any damage happened to the cell, that damage will be reconstructed by the process called as fibrosis process. And ulceration, so it may be acute inflammation, maybe histamines and leukotrienes and all the inflammatory mediators like macrophages which are involved. They may enter into the they may stase, they may cause fighting against the bacteria, or fighting against the various cell injury agent. Okay, so it may cause damage the mucosa of the one particular affected area, as it is, as a part of it is entering into the antigen which is present in that particular area. So sometimes the inflammatory compounds or inflammatory mediators may damage that particular tissues. And fistula, so they may form one new connection, the new connection in between the two organs, a new connection within the lumen type. Okay, so fistula is a one type of a lumen-like structure which will be present, which will be formed during the inflammatory by the new cells, which are formed, which are observed in the inflammatory condition, which is just like a tunnel-like structure to leak out the organ contents into the out of the body or into the new place of the body. So like anal fistula, vaginal enterovaginal fistulas are some of the things which are present, which are occurred in between the two nerve cells. And suppuration. Okay, whenever the bacteria are pyogenic in nature, which are able to produce the pus cells, which are able to convert the cellular material into the pus, which have the neutrophils, which have the debris, the dead cell debris. Okay, so it may be inflammation maybe leads to the supination, when the cell death may take place by means of necrosis. So that necro tissue, what are the debris contained in the necrotic tissue? What are the contents of the cytoplasmic content of the necrode cell? Cytoplasmic content of necrode cell, which are present in the form of cell debris. So they will be converted by the neutrophil cells, converted by the bacterial enzymes, or by the various local enzymes. Okay, digestive enzymes will be converted to the pus. Okay, so finally they are observed in the form of abscesses. Abscesses, it is just like a cavity-like structure, or it is a vascular structure which is filled with a pus. And scar formation. So acute inflammation is a one of the healing type, one of the step in the healing process. So as and when there may be a chance of healing. Okay, there may be a complete removal of any infectious organism. So whatever the damage happened to that infected cell will be replaced, or infected organ will be replaced by the new cells. And that will be formed the little different cells from the surrounding organ cell. That will be called as scar formation, or sometimes it will be leads to the chronic inflammation condition. Okay, so chronic inflammation. So whenever acute inflammations are not possible, overcome the stress conditions. Acute inflammations are not enough to remove the stress conditions. Okay, at that particular time, okay, there may be a recurrent acute type of inflammation. There may be a initiation of new steps for the recovery, or new step for the removal, or new step for the repair. New steps are required for the destruction of the damaged cell. If it is not destructed, that damaged cell will causes for the destruction of other healthy cells. So when such a acute inflammations are not enough for the maintaining of the repair of the cell, or maintaining of the destruction of the cell. Okay, there is a prolonged or continuous ongoing inflammations are takes place, that will be called as chronic inflammation condition. When acute inflammations are not sufficient, when there is a recurrent attacking of acute inflammation, and where there is a de novo, when acute inflammations are bypassed by any reasons, okay, at the initial stage, the body is not recognize the cell damage or cell injury. Okay, so immediately, without producing non-specific defense, they will produce a specific defense. Okay, and which will be extend for the longer duration, will be called as chronic inflammation process. So there are two type of chronic inflammation process based upon histological features which are observed at the end of the inflammation. There is a chronic non-specific inflammation. There is a granulomatous inflammation. Non-specific inflammation, chronic non-specific inflammation. Okay, there is a based upon the type of the cell, and type of the cell injury, type of the organism. Okay, there is a appearance of the cell at the end stage is not specific. And the granulomatous, there is a presence of granuloma at the end. Granuloma cells at the end of the cell repair. There is a presence of granuloma at the end of the cell repair. So when the immune cells are going to form like a wall-like structure, at the end stage, and that will be called as the granulomatous type chronic inflammation condition. Okay, so what are the basic cells are involved? So basic cells like macrophages, like activated macrophages and modified type macrophages, interleukins 1, TNF alpha, arachidonic acid metabolites, and growth factor, platelet-derived growth factor, fibroblast-derived growth. Fibroblast cells are very, very important cells which are present in the wound healing. Okay, wound healing, because the inflammation is one type of repair process, and lymphocytes, activated lymphocytes, and TGF beta, okay, and PDF, platelet-derived growth factor, transforming growth factors, and fibroblast growth factor derived. So interferons, okay, it is a activated macrophages. So and all these type of cells will be involved in the chronic inflammation mediated condition. And how this chronic inflammation will be identified. Okay, so what will be the difference between acute and chronic type inflammation condition? So when you have a fever, and when you have a severe anemia condition, okay, when there is a less number of healthy RBCs, called as anemia. Leukocytosis, when there is a reduction of WBCs. ESR, okay, elevated ESRs, erythrocyte sedimentation rate is increased. When there is a erythrocyte sedimentation rate increase, it's a one type of test, okay, clinical test done by the various microbiologists or various lab technicians to understand chronic inflammation conditions, and to understand some internal organ damage, elevation of in the erythrocytic sedimentation days because of prothrombin, entering of prothrombin in the bloodstream, okay, to act against the inflammation condition, to act against the repair condition. So whatever the blood vessels which are present in the circulation will be stick, RBC stick one with another, will form a very huge heavy mass, and whenever you are using in the erythrocytic sedimentation too. So within shorter time, the RBCs will deposit at the bottom. So elevated. So at that particular condition, so fastly the RBCs are get deposited. So elevated erythrocytic sedimentation rate, more erythrocytic sedimentation rate. Erythrocytes are sedimented at a faster rate. And amyloid, so presence of amyloid protein. Amyloid protein is a one type of abnormal protein produced by the various biochemical reaction, and which is going to cross-link the various structures. So at that time also they are going to affect the which will be easily found that particular edema conditions. Okay, granulomatous, just granulomatous is nothing but a formation of granuloma. Okay, or whenever there is a chronic inflammation, more and more inflammatory mediators and T cells and B cells, okay, all the inflammatory cells will go and form a type of wall-like structure. Wall-like structure against the inducing pathological organism. So that will be called as a granulomatous. That's it, not it. So this is the pattern of the process. Okay, which will be the first one? Okay, what will be the second one, and that all. That's it. Okay, and what happened? How the release, and what are the various inflammatory mediators will release in the inflammatory process? Okay, so not and yeah, one more thing here, we should be very careful. Biological oxidant. So biological oxidants are very important compounds involved. Very important compounds involved in the what you call the repair process. But these biological oxidants are very important for damaging of the various cells. Okay. So I'll go a bit faster. These are the eicosanoids, PGE1, PGE2, F2 alpha. So these are the components produced by the arachidonic acid pathway. Okay, if it will start COX pathway, so prostaglandins and thromboxanes are produced, and if if eicosanoids are entered in the lipoxygenase pathway, so there is a production. There is a production of your, but there is a production of compounds like leukotrienes. Okay, so repair and healing process, we'll discuss this in the later. So let us have a small look on bits related to the cell repair process, then we'll finish this thing. So 2 minutes we will finish it off. So we'll finish in discussion related to inflammation and repair, then we'll finish. So give an example of inflammatory mediators from the following. So histamine, prostaglandins and leukotrienes, all are inflammatory mediators only. Okay. Then in wound healing, process of wound contraction is due to. Okay. So as I said, the fibroblast. So fibroblast cells are very important cells. Okay, which are involved in the wound healing process. Okay. So myocytes. So myocytes are the basic cells, and fibroblasts which are going to produce the collagen content, and myofibroblasts, so which are also very important cells involved in the wound contraction process. Okay, skeletal muscle fibers, so they are no more involved in the fibers. So myofibroblast or fibroblast. Okay, so construction is only done after formation of the collagen fibers. So collagen fibers are formed by means of the fibroblast cells. Okay, so correct answer is fibroblast. So in inflammatory reaction, the term red indicates the redness. Okay, swelling is different, eating is different, and pain is different. So the rubor is indicates by the redness. And the meaning of phagocytosis. The meaning of phagocytosis is nothing but cell eating. So cell eating called as phagocytosis. Cell drinking is called as pinocytosis. Cell drinking is called as pinocytosis. Cell digestion Okay, it is a internal process. Cell digestion is a digestion done by the various enzymatic substances. Okay, then metastatic calcification. Metastatic calcification, when the calcium produced in the normal cells, calcium produced in the normal cell in hypothyroidism condition, hypoparathyroidism condition, in hypoparathyroidism condition. So that condition, when the calcium in the hypoparathyroidism condition, so when there is a resorption of the bones, okay, resorption of the bones and decalcification of the bones, or when there is a huge serum serum calcium concentration, or the serum calcium concentration is more, that at that particular condition the calcium is going to deposit in the normal cells. So that will be called as metastatic, okay, metastatic calcification. If it is deposited in the dead cells, in the dead cell, that condition is called as dystrophic calcification. So dead cells aithe dystrophic calcification, then normal cells means it is a metaplastic, okay, metastatic calcification. So with this, okay, I am very thankful to everyone, okay, and we'll finish today's class. Sir, thank you, sir.